Thank you. Welcome. We are starting chapter 14 today. We did chapter 13 last week in 2 classes, and I'm hoping to do chapter 14 in the next 2 classes. So we're moving along. Part of the reason I want to move a little bit faster is so that we can wrap up the information and practice a lot of this stuff before the test. Because more than I think any other unit, this unit has the most problem solving of any of the units we do, which I think for a lot of you will be good and you will like it, and it'll be a little bit more fun than, just memorizing phases and understanding why mitosis happens. So, chapter 14 is an introduction to genetics. And some of you have done some of this before. If the word Punnett square rings any bells, or dominant versus recessive genes, that's a lot of what we're doing today. But whereas in a regular biology class, I might spend a week or more on what we call monohybrid problems and then move on to dihybrid problems where I would spend another week or more. We're going to do all that today. Welcome to AP. But y'all can handle it. I think you'll be fine with it. Our story starts with an Austrian, not Australian, Austrian monk named Gregor Mendel. And I will say the name Mendel a lot. And the kind of genetics we're discussing today is often referred to as Mendelian genetics. It's the basic stuff that works on the simpler playing field. Next class, we'll talk about kind of some of the weirder examples that aren't really that weird. They're just a little bit different. Mendel, as a monk, spent a lot of his life in a monastery where a group of people that dedicated their lives to their religion brewed their own beer, sold it, grew their own food, and also sold it, and were completely self-sufficient. And he spent a lot of time in the garden, and he took that opportunity to make probably the original breakthrough in genetics. And he did so using pea plants. So he grew peas, and he crossed them, meaning he bred different pea plants together and recorded what would happen. And this was an excellent organism, because remember, a pea plant is a living thing. It's a plant, so it's in its own kingdom, Plantae versus Animalia, but plants also have sex. They produce pollen, which makes sperm. They have flowers that are female and produce eggs, and they get their pollen to the female flowers, and sexual reproduction occurs. Now, since a plant cannot move to its mate, how does it get its pollen, the male gametes, to the female gametes? Wind, which I mentioned in the first lecture of this semester, is the way some plants do it, like pine trees. But for most flower-producing plants, they produce nice-smelling, sweet-tasting flowers because the birds and the bees. The birds and the bees are what help plants have sex. So that's where that comes from. Now, as with my lemon tree that is in the greenhouse, birds and bees don't get in there very often. Sometimes a bird gets stuck in the greenhouse. Sometimes it doesn't make it out. So I actually help my lemon tree with sexual reproduction by simply touching different flowers with my finger, and I just play the role of bee. And you simply just swab a little pollen on your finger and swab other flowers with that pollen, and the plant takes care of the rest. And baby plants are born. Now, in modern plants, not say moss or ferns, but in most of the plants that are on the evolutionary scale more recent, where do they put their baby? What structure holds the baby?
Seed.
The seed. The seed is where you find the embryo, and it's what provides the food. Now, this is not in your book for this chapter, but I'm just giving you a basic idea of a plant's lifestyle so that this experiment that we're going to talk about in a second makes sense. So when you eat your peas, your green peas, or your yellow peas, there are yellow peas, or nuts, or any other seed or nut, you are eating the plant's babies. And a substance called the endosperm, which is basically the meat of the nut that provides all the nutrition for that baby. Because the plant doesn't have a womb where a baby develops. That's the function that the seed plays. Okay, so here's the deal. Mendel noticed that his pea plants had a lot of different traits that were somewhat like this or that. He could grow peas that were green or peas that were yellow. He could grow these round, smooth peas or these weird kind of wrinkly peas. He had some pea plants that grew to a normal height, and then he had some pea plants that actually were suffering from dwarfism, where their growth would be stunted fairly early on. He also noticed that his pea plants sometimes produced purple flowers, and sometimes they produced white flowers, which is where this experiment comes in. And this is probably your first true genetics experiment that's at least recorded and considered to be a serious genetics experiment in the history of genetics. He took two what he called true breeding strains of pea plant. He took pea plants that had purple flowers, whose parents had purple flowers, whose grandparents had all purple flowers, and I think even went back that fourth generation. I forget how many generations it takes to have true breeding, to have that definition fulfilled. But he basically took purple flowered pea plants that had come from a long line of only purple flower plants. He then crossed them, and you'll see this X here. It just means he bred them or mated them together with pea plants that had only come from a long line of only white flowered pea plants. So these are what we call purebred or true breeding characteristics. And this generation is called the pea generation, referencing the parents. These are the original parents in this experiment. And we call it the P, or P1 is sometimes the terminology used. Now, he took all the peas, which are the babies, the offspring, the seeds of these plants, and he grew all of them into adults, and then looked to see if they had purple or white flowers, or a blend, or flowers that were purple and with white spots on them, or whatever. And what did he find about the children of these purple, white flower plants? What were the children like in terms of flower color? They were all purple. Very good. every single offspring from this cross came out to have purple flowers. Not a single white flower. This is what we call the F1 generation. The F word here is filial, which references like family, but in this case kind of means the children, the first generation you produce from the original parents. We also use this term hybrid, which is, I think, kind of a corruption of the phrase half-breed, which is kind of a phrase to use. But hybrid is kind of the scientific term for your parents had came from two different strains, two different genetic types. So this hybrid generation were all purple. Now, in animals, this doesn't work that well. You can get problems with inbreeding, but plants actually have no problem with inbreeding. In fact, some plants produce flowers that have both the male and female parts in the same flower. So a plant can actually fertilize itself in some cases. So plants are very open breeders in this sense. So what he did was he allowed this generation to only interbreed with itself. And what do you think the next generation showed up as? How did it look?
Purple.
It was mostly purple, but there were some white flowered plants. The white flowers reappeared after skipping a generation. And you've probably heard that phrase before when it comes to some traits that run through families about like, oh, it skipped a generation. Oh, my grandfather had that characteristic and I do, but my parents didn't, so it skipped a generation. This is, once we explain what's going on here, an example of that. Now, in this example, he did this with literally tens of thousands of pea plants over his career. I cannot imagine how much work it was to write all that down. And the monastery burned like 40 years later, and almost all his original books were destroyed. But we still have the data from other sources. But in this case, we have 705 to 224 purple to white as a ratio. And using whole numbers, what's that rough ratio reduced to? It's somewhere in the 70% range. The answer is kind of staring you in the face. Give me just a simple single-digit whole number ratio. 3 to 1. Now, this is not a perfect 3 to 1, but we're going to see that a lot in this unit, where you have to kind of come up with a rough estimate as to what the simple ratio would be. But if you repeat this experiment, you will typically get around 3 to 1 purple to white. So, if you're not familiar with genetics, if you didn't do some of this stuff in like 7th or 8th grade, I do want to take a moment to mention that at this point, Mendel realized that one of these traits was showing up more and preferentially over another, but he didn't know why. Remember, it would be 70 years before we discovered the structure, the double helix structure of DNA, and even understand that there were such a thing as genes or genetics. So he was trying, he was totally in the dark here. But he was able to recognize that purple flowers in this organism were the dominant trait, and the white flowers were the recessive trait. So this is where the idea of dominant and recessive comes in. Now, to flash forward to today, How do you explain this? And I know this is a really busy diagram with a lot going on it. But here we have the original purple and white parents for the flower color of these pea plants. And I wouldn't bother trying to read everything, but here's the deal. In the case of pea plants, the color of the flower is controlled by a single gene. Your skin color is actually controlled by like 6 genes. So we have lots of different shades and hues of skin color in the human population because there's a lot more genes that are involved with it. But in pea plants, for the color of flowers, you have basically just a purple gene and a white gene. And just like in humans, pea plants have 2 matching chromosomes for every chromosome. So on one of your pea plant chromosomes, you have a gene for purple. And it's hard to see, but in your book, when you do your reading, you can look at this again. There's a little purple segment right here on this chromosome that represents the section of DNA that tells the plant, basically, to make a purple chemical that makes the flower purple. So there's a purple gene. Now remember, this plant was true breeding. So both of its chromosomes, one from one parent, one from the other, were both containing or holding a purple gene. And the same was true for the white flowered plants. Their matching chromosomes for this one chromosome, the homologous pair, both of these chromosomes had white flower genes at that location on that same chromosome. And this is how it works for humans for all our traits. It's just not always this simple. Now, through what process do pea plants and humans make sperm and egg cells?
Sexual reproduction.
Well, we fuse them together to make babies in sexual reproduction, but what process actually makes the gametes? Meiosis. That's why we covered chapter 13 last week. So the pea plant in the male or female part of the flower, which that's all flowers are. Flowers are the sex parts of plants. I don't know why we give them to each other, but it's kind of weird if you think about it. But in the sex parts of plants, which are the flowers, you have cells that undergo meiosis, and in this case, they're saying that the purple flower is the male, the white flower is the female. So we'll go with that example, but it doesn't have to be that way. The sperm cells that a purple male flower would make segregate out, they separate out, and every sperm cell that is made by this purple flower is going to carry a chromosome with a purple gene. Because that's the only gene it has on its chromosome for that specific chromosome. Same for this true breeding white flower pea plant. All the eggs she makes will have white genes at that location of that chromosome. So in their children, every sperm cell will carry a purple gene, every egg cell carries a white gene, and the children then have in their matching pair a purple gene and a white gene. Well, we will talk in a second about how when there's a purple and a white gene both present, the purple shows up, which is a situation where we say that purple is dominant, because when both are present, it's the purple that shows up, not the white. But why does white come back? Well, the sex cells of this generation, the sperm or the egg cells, can have purple or white genes that they're carrying, and occasionally a sperm cell from a pea plant with white flowers that's carrying that white flower gene, and an egg cell from a plant that was carrying the white flower gene if these two, this is a sperm and this is an egg cell, and they fuse, you only got white flowered genes present. And that's the only way to have a pea plant with white flowers. Okay, and we'll go through this more as we practice these problems. Now, I want to look a little deeper here. What is going on in the chromosome? Because there's a term that shows up on this diagram, and it confuses people sometimes, and the term is allele. You see the word allele here. The word allele is almost the exact same thing as the word gene. It's just a little bit more specific. So a lot of times, and I mean honestly on a test, almost any question, I could use the word gene or allele and it kind of wouldn't matter. But I do want you to know the difference. Gene is a general term for just a segment of DNA that codes for a certain trait. Allele is the flavor. Is it, you have a flower color gene. Is it the purple allele or the white allele? That's kind of how you use that phrase, allele. So what is a gene, or more specifically, an allele? Well, here we have a pea plant chromosome. I don't remember how many chromosomes are in a pea plant. In humans, the number is 46. We could pretend it's that same number. It could be 30-something, 50-something, who knows? You'd have to Google it. But here's a pea plant, one pea plant chromosome with a gene for purple flowers. That gene is just a segment of DNA. All right? So if I ever say gene, I mean a section of DNA that is 50 to maybe thousands of bases long. So in order for a pea plant to make purple flowers, that segment of DNA in that chromosome has to say CTAAATCGGT and on for at least probably 50 to 100 letters. It takes a certain number before you can actually have a gene, but it kind of depends on the characteristic or trait. The white allele, in this case, they are showing one change. One letter changes and you get a different trait. Now why? Well, DNA tells ribosomes what proteins to make. And in the case of pea plant flower color, this purple gene, this segment of DNA, tells the ribosomes of the pea plant to make a specific enzyme, and that enzyme drives the production of a purple pigment that ends up in the flowers. And that's where the purple comes from. Now, and this happens in humans as well, there is a mutant that just can't make the purple. So it shows up as white. It's also oddly where white people come from. If you're a white person, you have changes in your DNA that prevent you from making as much melanin in your skin cells. So you just don't have as much melanin and you're a much lighter shade of brown. So what happens, and this explains actually the dominant versus recessive concept in the purple versus white flowers here. A plant is purple if it can make this enzyme. Well, all it needs is one gene out of the two to make that enzyme, and it will be purple. The only way a pea plant makes a white flower is if neither gene is capable of making this enzyme, And the only way you're going to have that is if both genes are the mutant version, which does not end up in color. Isn't that fun? Isn't that fun?
It's so much fun.
Good. Thank you. So, this is where we kind of start heading towards how we solve these problems or show this on paper. So again, this is the P1, the F1, and what we call the F2. Parents, children, grandchildren, 3 generations. This is our original purple and white, the hybrids, and then our 3-to-1 ratio of purple to white. How did this happen? Well, the purple genes, remember you got 2 of each gene. You have 2 chromosomes, each one with a gene or allele on the chromosome. These purebred purples had purple on the 2 chromosomes. The purebred whites had white. All the kids got a purple from one parent and a white from the other, so they were purple-white. Well, based on how this gene operates, if you have 1 purple, it's enough to make the enzyme that causes purple. So purple's the dominant, white's the recessive, which we see because they were all purple. What happens here then, in this generation, is the same thing as a coin toss. These purple-white flowers, which look all purple but carry a hidden recessive white gene, they could pass on in a 50-50 coin toss, either purple or white. The other parent, who is also big P, little p, as we'll say a lot in this unit, also could have a 50-50 chance of passing the purple or the white gene on. So we set up this Punnett square, which can be used for coin tosses or any other 50-50 scenario, where you have the possibilities of one parent's donation on the top, the possibilities of the other parent's donation on the side. Now in this case they have the father and mother. I don't know if that's a standard practice. I don't really care which parent's possible gametes you show here and here. But basically these letters represent what sperm cells the males could give, what egg cells the parents could give, and then the possible combinations that could show up in the children. And you can see in this generation you got 3-to-1 because there's a 25% chance of getting purple. Just like if I took 2 coins, 2 pennies, and flipped them both, there's a 25% chance they both come up heads. There's also a 25% chance that they both come up tails, which in this example would be the 2 white genes together to make the white flower pea plants. But then, just like with flipping pennies, you can have 1 heads, 1 tails, or 1's tails and the other's heads. Those are 2 different possibilities, so they add up to half the possible outcome, where you have half, or 2 out of 4, as purple gene with a white gene amongst those 2 chromosomes. Since purple is dominant, you see it 3 times out of every 4. Now, before I move on, I want to say something, because we're going to draw a ton of these Punnett squares over the next week or 2. And I don't want you to have this misconception. This Punnett square is not a magic 8-ball. It's not a seance board. It is not the oracle. If you know your blood type or your eye color, we can play around with like what your kids could look like depending on what your mate had as traits. We'll do some of that in here. But this is simply a statistical chart that shows you probabilities. It doesn't predict the future or tell you the future. So if you have two of these hybrids make 100 kids, they could all have white flowers. The odds of that are very low, and I wouldn't go to Vegas and bet on it. But it's just like flipping a coin. If you take a coin and flip it 10 times, how many times will it come up heads? No, not 5. Who knows? How do you know? Now, if I had to bet money, I would probably put it on 5, because those are the odds. But have you ever flipped a coin twice and gotten the same side come up both times? Yeah, you can violate probability. So it does not mean every 3 out of every 4 will be purple. It just means that you would expect around 3 to 1 as your ratio. This is just kind of a hand-drawn diagram that the book had. You will have to draw the squares and write in the letters and fill it out if you need to. Some people can do this in their head, and that's awesome. Now, there's a few terms that I have to go over. This is important vocabulary, and then I think we just have 2 quick slides after this. So it shouldn't take us 10 more minutes to get through this, and then we'll be done with this part of the chapter. But here's our 3 to 1 ratio from that 3rd generation, okay? And if you look at them with your eyes, just on the surface, it looks like 3 identical purple flowers and 1 white one. And we have a phrase for the physical appearance for any trait in any organism. And that term is the phenotype. And I will use the word phenotype a lot. Phenotype is the physical appearance. It's just what it looks like. So if I ask you about the phenotype, you would just look at the flowers and go, well, there's 3 purple ones and 1 white one. The phenotypic ratio is 3 to 1. And that's all Mendel could do back in the 1870s, 1880s. But once you know how the genetics works, and that there's 2 genes, and what's crazy is Mendel was able to take the data that he got. He took the numbers, and he knew that there was a purple something and a white something, and that it was working in the same way flipping a coin would work statistically. He didn't know what those somethings were. 80 years later, we knew those somethings were segments of DNA and chromosomes. He didn't know about any of that. He knew that plants had cells. That's as far as science had gotten at that point. But today we know that some of these purple plants are purple because both of their genes for that trait are purple. And some are purple because they have 1 purple gene, and that's all you need to have purple flowers, even if you have the other gene as a white gene. And the only way you can have a white flowered pea plant is to have 2 recessive traits stuck together, or 2 recessive genes together. 1 on 1 chromosome and 1 on its matching pair. These are called genotypes because they tell you what the exact gene combo is. This is the gene combination that in these cases actually result in purple, which is a phenotype. So with these kinds of questions, you could say, oh, my phenotype, I got 3 purple. But genotype, that's One purple is because of big P, and two of those three purple flowers would or expected to be caused by big P, little p together. If the genes are the same, we use the phrase homozygous. Homo means same. If you have different alleles, a purple, which is different from the white in the same organism, we use the prefix for different, which is hetero, and we say heterozygous. And I will be using these terms. These terms are used in your practice sheets. That's why you want to do these practice sheets I have on the coursework page for today. Because I'll use these terms, and you need to start practicing seeing these words and relating them. So that's 4 words. Phenotype, genotype, and then homo and heterozygous. Questions so far? Have you all had this? Some of it? It's familiar, I think, for a lot of people, which is good. Notice when we write the phenotype ratio, we would say something like 3 purple to 1 white. And then when you write the genotype, its ratio, which we call the genotypic ratio, you actually say like, well, I've got one that has 2 dominant genes, 2 that have a dominant with a recessive, and 1 that's got 2 recessive genes. And we would say homozygous dominant, heterozygous, and then how would I say the little pp? Homozygous, recessive. Good. Specify. Very good. Okay. Last 2 slides. The last one's got a little bit more information. But here, there is a concept your book talks about called the law of segregation. To segregate means to separate. So, for all your chromosomes, you've got 2 of them, right? When you make sperm or egg cells, you basically, any single sperm cell, any single egg cell will have a 50-50 chance of having one versus the other, all right? Works just like tossing coins. So if you have type AB blood, which we're going to talk about blood types later, that means you have an A gene and a B gene. So if your blood type is AB, when you produce a gamete, that gamete has a 50-50 chance of having either just A or just B in it. You separate your 2 chromosomes during anaphase, and you then place them into your sex cells, and it's random. So the coin tossing analogy is used a lot. But let's finish with something more complicated, but on the exact same train of thought. There is also a law of independent assortment. Your book also discusses this, so I would pay attention to it in your reading. And this is the last thing I want to talk about. And the idea of independent assortment is that for many, but not all, for many of your physical traits, it doesn't matter, say, which blood type gene you pass on to your child. That is independent from your eye color gene. Okay? Now, some traits do track together pretty well. Skin color and eye color, they actually aren't completely independent from each other. You typically see a lot of tracking where they stay somewhat together, the skin and eye color, but they don't have to. But for most traits, you would consider one trait independent from another when it comes to your chances of inheriting it or passing it on to your kids. And an example of this is, again, Mendel's pea plants. He did other experiments. I told you pea plants can be yellow or green. I don't know why all peas that you buy in the store are green peas, but there are yellow peas out there and other colors as well. Well, Mendel found that yellow is actually dominant to green. And so we use a capital Y for yellow, just like we used a capital P for purple. By the way, we did this with purple and white. The green gene in pea plants is not a G. We use the lowercase version of whatever letter we choose for the dominant gene. So big Y is for the dominant yellow. Little y is for the recessive, whatever it is. Oh, it's green, cool. So big P was the purple gene symbol. Little p is actually the symbol for the gene for white flowers. And this is part of getting the hang of these genetics problems. You will also find that round, smooth peas are dominant to a wrinkled, kind of not so smooth pea shape. So we have capital R for round, and then lowercase r for the recessive wrinkled. And you can look at both or any two traits at the same time. And this is where genetics problems start getting a little bit more difficult. In this case, we have a purebred, true breeding yellow and round. It's homozygous dominant for both. So we would write it as big Y, completely dominant, purebred yellow. Big R, completely dominant, homozygous dominant, purebred round. The only way you can have a wrinkled green pea is to have all recessive, because green and wrinkled are the recessive traits, so you have to be homozygous recessive to have them. When he bred these two together, or if anyone were to breed these two together, the only genes that this pea right here could pass on would be the dominant ones. The only genes that this pea plant could pass on are the recessive ones. So the child, which we actually call a dihybrid, has dominant recessive, or we say heterozygous for both. Now, what does this pea look like? Well, it's drawn for you. It's yellow because yellow is dominant, and it's round and smooth because round and smooth is dominant versus wrinkled. So just like you took purple and white and got all purple, if you take yellow round and green wrinkled, you get all yellow round because it's the dominant one. But then what I would do is ignore this. This is the incorrect hypothesis. This is what we are going to look at. Where it gets tricky with these problems is you've got two individuals that are both heterozygous for both traits. So what you find is that both parents look like this. So this parent can give both dominants, the yellow and round, or it could give, in its sperm or egg cells, a yellow with a wrinkled gene. Big Y, but little r. Or it could give green and round. Little y, big R. Or it could give both of its recessive ones. Little y, little r. And the other parent can do the same. And if you are looking at two traits at the same time, it's a 4x4 Punnett square, and you have to combine all these letters together to get the possible genotypic and phenotypic ratios. So in this case, if you reduce it, it still works out pretty well. Like in this specific case, you actually still do get a 3-to-1 for both of these traits. But if you look, this one's homozygous for both. This one's homo but hetero, hetero but homo, hetero, hetero. And as you go through, you find all sorts of possible genotypes. So we're going to play around with this. and I will help you with it. Because this is where people, the 2x2 Punnett squares, people sometimes have a little trouble and they learn them real quick. But then when you get in these 4x4 ones, which you need to know how to do, it gets a bit trickier. I will probably not be lecturing quite as much the next class or two. I hope not. This was a lot to go over with you. And I want to use the time we have over the next 2-3 classes to practice a lot of this. So on your coursework page, there is a task that is, I think, just genetics practice, something along those lines. And there's 2 documents linked. One has the simpler 2x2 Punnett squares, and then the other one deals with these more complex 4x4. You can print them out, and I'm telling you, let's use the time we have to try them, and I can help you with them. So reach out to me on Google Meet or office hours or in class or whatever, and we can work on those together. You can stop the video. Make sure you do your reading.